Thank you so much for joining us. Today we'll be asking you some questions to get a better understanding of some of the courses you teach, the investigations you conduct in the lab, and the thesis projects you supervise with fourth years here at Western. To start, maybe tell us a little bit about yourself, what got you into science, academia, and where you are today.
Okay, a little bit about myself. I guess originally from just north of Toronto, Thornhill, Ontario. What got me into science, I always loved science in high school. I actually tended to like the chemistry, physics, math a little bit more than biology. So when I was in high school, I actually never took biology. And when I came to university, I was almost an engineer actually. When I came to university, I changed last minute to go into science and took my first biology class and found out that I actually liked the biological sciences more than the physical sciences and sort of switched my path there. Had no idea what I wanted to do with my life. I just knew that I liked science, so I started it down that path, and really was taking a pharmacology class in third year that really let me know sort of what I like to do and sort of pursued that, and the rest is history.
So could you briefly tell us what you and your lab are investigating?
Sure, yeah, happy to. So we're really interested in sort of why people respond differently to medications, specifically in the area of kidney disease. I guess one particular project that we're working on right now is why do some patients have kidney toxicity taking certain drugs when other patients don't? And sort of figuring out why that is, we hope can prevent those patients that do get kidney toxicity from taking some medications to basically prevent it.
So what's one piece of equipment that you use in your lab, and how does it relate to your research?
Yeah, well, if you ask the people in my lab, bar none, the most frequently used piece of equipment is a liquid chromatography system that's coupled to mass spectrometry. We use this to measure drugs, metabolites of drugs, and all sorts of other small molecule metabolites, and we use it to look at those drugs and metabolites in all sorts of different biological fluids, plasma samples, urine samples, tissue samples. We use it to measure things and do assays called metabolomics, where we're looking for metabolites that might predict why certain patients respond a certain way to different drugs. We really do use that piece of equipment quite a lot.
All right, so which courses are you currently teaching this semester?
This semester I'm involved mostly with the Scholars Electives program and Medical Science 1000Y. So Medical Science 1000Y is a new course in medical sciences that just started this year that sort of allows students an exploration into the different areas of medical science. I think what we're finding is that students in the program are coming in not really knowing the options that are out there and sort of what they want to study, and this course allows them sort of to take a tour through the different offerings in the medical science program. So tonight actually is pathology will be coming to talk about their program and module. I'm also pretty heavily involved with the Scholars Electives program this term, and that's a program where students get involved in research and community engaged learning throughout their undergraduate journey. So doing a lot in terms of organizing those courses and the research project presentations and grading, etc. Last semester I was teaching in human pharmacology and therapeutic principles. That's pharmacology 3620, which is actually kind of nostalgic for me always, and I tell the students this, that it's the course that I took as an undergrad student, because I did do my degree at Western, that got me interested in pharmacology. So it's kind of crazy that I'm teaching in that class now. I also teach in pharmacology 4660A, which is human toxicology, a fourth year advanced toxicology class. So I teach in that, and I teach a little bit to the dental students about pharmacology and principles of pharmacology.
So could you describe or give examples of the type of work or type of project that an honor specialization student would do with you in fourth year?
Yeah, sure. I can go back to last year and our current students who are in the lab this year. So the projects that I was talking about earlier about the drug toxicity and precision medicine, we've had students working on those projects looking at metabolomics on human urine and plasma samples, trying to figure out what metabolites predict who's going to get drug toxicity and who's not going to experience drug toxicity. We've also in our lab recently identified sort of a new metabolite where there's not very much information known on it. It's called TMAP, and one of the undergraduate thesis students this year is working to learn a little bit more about TMAP and how it functions in the body. And I guess we also do a little bit of work sometimes with small companies, and there's a company project where we're looking at different areas, sort of doing a little bit of work in collaboration with the company that one of the undergraduate fourth year thesis students is working on.
All right. If there were one or two tips that you could give to your students looking to excel in your classes, what would you say?
Yeah, I think I talk to a lot of students, and I think the thing that gets to a lot of students is the workload in university compared to high school. And I think as students move through the programs, they really find what works for them. But I think really the time management is probably the most important thing to get a good handle on early on, to do things in advance. Everyone has that sort of quieter time, and to be productive in those times and not leave everything to the last minute. I think that's easy to say and it's hard to do. I think everyone sort of works to deadlines, but if you can dedicate a little bit of time every day to just be going over things, it makes your life a lot easier in the long run.
So, what would you say are the top 3 things that you look for personally in a prospective thesis student?
Yeah, I mean, the number one thing for sure is, I think the students that approach me to work in the lab are coming from our BMSc program. They're all smart enough to work in the lab. I haven't encountered a student yet in all of my years that's not smart enough to be working in the lab. It really, for me, is the personality. It's trying to find the right fit. It's a team environment, and wanting to make sure that people can work well in a team. And I think the other thing is just probably evidence of work ethic and experience in making sure that people are dedicated to doing what they want to do. Research is not a typical 9 to 5. There are times where you're really busy. There's times where you're not as busy. So I think work ethic, and also, again, time management, like really being able to balance your schedule and get the work done when it needs to be done, and also maximizing that time when it's quiet.
So, what has been the favorite part of your research? Is there a particular moment that stands out for you?
There's so many in research. There's so many highlights. It's a long journey. It doesn't work out every time the way you expect it to. So when you hit something that really works out, or you publish a paper, it feels like a really monumental win. I would say, for thinking back, recently, the identification of that TMAP metabolite has probably been one of the more exciting things that's happened in the lab. It's sort of something that we didn't know anything about before finding it in one of our metabolomic studies, and there's really little to no information about it in the literature. So it was really exciting to find that, and it opens a lot of doors for the lab. It also presents challenges, because usually when you want to study something, you read other people's papers to get the background. You go to a vendor to purchase the material, and we couldn't do that at all with this. It's completely new, but that's also really exciting.
Awesome. And what has been maybe a really challenging moment, or
Yeah, so I mean, there's with the successes, you read someone's paper and it looks like it could be done in like 3 months, that you could just do it, but really there's so many challenges and setbacks and things that are going to go a certain way in your head, and then they don't. So, with the TMAP project, for example, just having to as I mentioned before, not knowing anything about it is a challenge, but also, there's all sorts of things that are challenges, but they're all overcomable challenges. When we talk about infrastructure, we have really sophisticated infrastructure here, but like anything, it has wear and tear, it breaks down, it needs repair. So sometimes you have this big experiment planned, and you have to wait on the repair of the infrastructure before you can do it. So there's lots of little roadblocks, but you just have to keep a positive mindset. They're all overcomable, and you just move on, and it's all part of the process, troubleshooting too in research, right? It's what makes you be able to tackle a problem a little bit better, experiencing those setbacks.
All right, so 2 quick questions for you at the end. First, do you drink any coffee, and if so, what's your favorite type?
I do drink coffee. I love coffee. I last Christmas I got an espresso maker in my house, and it's a game changer. I love an Americano, a black Americano. So still playing around with how I like it best, which is it's like a little science experiment every day, right? For how you grind it and all how much coffee you put in. So love the Americano.
And secondly, what was the last time you broke something in your lab?
Ooh, yeah, I'm glad the students aren't around to hear this one. Everybody breaks things in the lab. The reality is, in my career, I've like transitioned into an administrative role, so I spend less time in the lab than I used to. So I actually can't remember the last time I like broke some glassware. So it's been a while, but that's more reflective of the fact that I'm not in the lab as much, rather than my dexterity or skills at handling all the glassware and everything.
All right. Well, thank you so much for your time, Dr. Urquhart. I'm sure your insight will be greatly appreciated for a lot of these first and second year students.
Thank you.